Echinococcal infection is distributed in South America, Europe, southern and central Russia, Central Asia, Australia, and Africa. The risk of infection is highly influenced by the extent and nature of human association with dogs. The parasite is widely prevalent in regions of the world where dogs are used to care for large flocks of sheep. This map illustrates the global distribution of both echinococcal species, with E. granulosus represented by regions marked in black and E. multilocularis marked with an X. The relationship between Echinococcus and its intermediate and definitive hosts is relatively straightforward. Adult worms inhabiting the small intestine of the definitive hosts release eggs in the stool. These eggs are then ingested via fecal-oral transmission by the intermediate hosts, allowing for the eggs to hatch and larvae to penetrate the intestinal wall. Once inside, the larvae are taken up in the bloodstream and distributed to the liver, lungs, and other organs, where they form hydatid cysts that enlarge slowly over time. Definitive hosts become infected when they ingest these cysts, for example, a dog consuming the infected viscera of a sheep, allowing for immature protoscolices from the cyst to attach to the small intestine of the definitive host and mature into adult worms, completing the life cycle. Hydatid cysts are composed of 3 layers. The paracyst is the outer layer, consisting of modified host cells, fibroblasts, giant cells, and eosinophils, which together form a rigid protective layer and represents the host response to the parasite. The exocyst is the middle laminated membrane of parasitic origin, permitting the passage of nutrients from the host to the parasite. Disruption of this membrane predisposes the cell to degeneration, death, and bacterial infection. The endocyst is the inner germinal layer. Immature protoscolices multiply within outpouchings of the germinal layer, known as broad capsules. Daughter cysts, smaller hydatid cysts derived from the primary mother cyst, may also develop within the endocyst. This photo shows the typical appearance of the laminating membrane of a hydatid cyst which has been removed surgically.